localized phylogenetic tree, but it's not just based on 16S ribosomal RNA. I'm sorry. Thank you. It's based on the sequences of different genes and different proteins. You might remember when we were talking about this in class, I said just using, it would actually be 18S ribosomal RNA for eukaryotes, but just using ribosomal RNA sequences is not enough for showing biology for eukaryotes. So if you're interested in which groups, you can find them on here. And I believe I posted this for you. I'm going to have to get the rest of these ones out to show this. One of the organisms you're going to look at is Exploidia. And this one's kind of a fun organism to look for. One of the things you'll notice is that the nucleus of the organism usually has kind of a delicate, kind of definite periphery if it's well stained. Usually the cells are oval to circular in shape. This is a protozoan that causes amoebiasis or amoebic dysentery, gastrointestinal illness in people. This particular amoeba, antiamoeba, has ingested some red blood cells. I've never seen one that ingested red blood cells on the slides that we have for you to look at. So chances are you're just going to see a cell that's kind of oval shaped that has a prominent nucleus. This organism also has a cyst stage, and sometimes even though the slide might be labeled for the protozoan, you can also find cysts. Something that looks like that slide over there, that would be the cyst stage. The next group I want to show you includes some apicomplexans. And the particular organism is Plasmodium that you're going to be looking at, and I believe you have Plasmodium falciparum. This is a parasite that infects red blood cells. So what you have to look at are red blood cell smears. So you're going to be looking for red blood cells where it looks like there's something inside. And this slide is just showing a series of drawings of all the different stages that this particular organism can exhibit in the red blood cells as it goes through its cycle. So you may see some like this, where the parasite looks kind of like a ring. You may see others where it's occupying a greater, volume, a greater proportion of the red cell. Or you might see some of the other stages here. Here are some actual photographs of smears. So here's a cell that's not infected. Here's a red blood cell that is. This one's not infected. This one is infected. So you kind of get the idea. Now to look at these, if I were you, I would try to scan the slide. You get it in focus on low and then scan under the high drive. But you'll have to use oil on this one. You should be able to find, I think, these on low power, but you can use oil to get a closer view. No, you won't find them on low power. You get in focus on low power, and you can scan on high drive, and then, hopefully then focus on the oil. So you won't be able to see real well on low. All right, that's just another organism. We have, I don't have slides of this organism for you to look at that are well stained. I just put it in here because it has such a distinctive appearance. It causes this causes the disease called giardiasis, or sometimes called beaver fever. Anybody heard of beaver fever, maybe? Anyway, the reason I put it in is because it looks like a little face and has two nuclei, and it's kind of neat once you see it and you don't forget it. I forgot to mention this for Plasmodium, that causes malaria, of course. It has stages that occur in the red blood cells and stages in the liver. You have slides of this. If I were going to start with something, I would start with this, because you don't even need oil to look at this. High dries plenty. This organism is large. It's got very distinctive appearance. It's well stained with multiple flagella. Causes vaginitis in women. sexually transmitted. And I think, oh, I should have had one more. Must have deleted something. Yeah, my trypanosomes are missing. So, the trypanosomes look kind of, they also should be pretty easy to recognize. You should have trypanosome cruzi, and they kind of look, maybe kind of like that, and they're flagellated. So hopefully, and it's a pure culture, so you won't have to be looking for a lot of stuff. The challenge with looking for the entamoeba is that you're going to have a lot of debris, but it's not really, because usually those specimens are prepared from fecal specimens, so there's going to be a lot of other junk in there, despite the fact that they process it. I'm going to leave you to it. There's one slide set for each pair. Microscopes are there, and lens paper and things like that are in the little blue top container. And I have paper for dry.